Hi, everyone. Thank you so much for tuning into today's Author Talk series brought to you by the Library Speakers Consortium. The Library Speakers Consortium is a partnership of more than 500 library systems across the US, Canada, and New Zealand with the goal of making author talks more accessible to communities of all sizes. I am Brandon Adler, your host and moderator in conversation with each of the authors and presenters you will hear from in our series. Today, we are so honored to have Smithsonian scientists, Dr. Brian Gratwick and Dr. Gina Dellatonia with us to talk about their research and how it relates to the book, Extinctopedia, Discover What We Have Lost, What Is at Risk, and How We Can Preserve the Diversity of Our Fragile Planet. Before we jump in, I want to give you all that standard housekeeping information that keeps everything running smoothly, and then I'll turn things over. We have compiled some questions that were pre-submitted with registration, but we welcome you to also submit questions during the presentation by clicking the questions button that's below the live stream video. There are quite a few of you tuning in today, and we will take as many questions as we can during that Q&A period. So definitely remember to submit any questions that you have. As a reminder, there will be a link to the recording sent out to all registrants in about 24 hours after the end of the event. Now to formally introduce our guests for today. Dr. Brian Gratwick is a conservation biologist and leads the amphibian conservation programs at the Smithsonian's National Zoo and Conservation Biology Institute. Brian's focus is building capacity to conserve amphibians in Appalachia and Panama, developing outreach and educational programs and exhibits to build public support for amphibian conservation, and research to develop tools to reintroduce amphibians back into the wild. Gratwick grew up in Zimbabwe and began his conservation work researching the impacts to freshwater fish in Africa. In 2000, he was awarded a Rhodes Scholarship that took him to Oxford, where he studied marine fish communities in the British Virgin Islands for his doctorate. After completing his doctorate, Gratwick moved to Washington, D.C., where he worked as a program administrator and then assistant director to the National Fish and Wildlife Foundation's Save the Tiger Fund. Dr. Gina Dellatonia is a researcher and conservationist, currently serving as the executive director of the Amphibian Survival Alliance and a research associate at the Smithsonian Tropical Institute. Dr. Dellatonia earned her PhD in biological sciences with a specialization in molecular and cell biology from the University of Maryland. Her groundbreaking research focuses on the reproductive biology of amphibians and the development of assisted reproductive technologies for endangered species facing challenges in reproduction. Her dedication to amphibian conservation extends to her involvement in global, regional, and local initiatives where she fosters collaborations and spearheads innovative projects to safeguard amphibian populations. Her expertise and passion have made her a leading figure in the field of conservation biology, inspiring positive change for the future of amphibians and their ecosystems. Thank you both so much for being here. I'm going to turn things over to you now.
Thank you so much. Welcome, everybody. Good morning, afternoon, everybody. First of all, thank you. We want to thank our host today for inviting us to discuss this amazing book. And thank you, everyone else, for joining us today. We're excited to take you on a journey through the content of this amazing book, exploring its profound insights and significance in the context of extinctions and conservation. Our talk today is titled Extinctopedia, Learning from the Past to Shape the Future of Conservation. We hope to inspire a meaningful conversation and reflect on the critical role we all play in preserving our planet's biodiversity. So let's dive in. So this is the beautiful cover of the book, and Extinctopedia is not just a catalog of extinctions. It's an emotional journey through stories and species that have vanished or are in the brink. It uses vivid illustrations and accessible language to reach both youth and adults. I want you to pay attention to the quote on the bottom of the slide by Joel Sartori. And this quote reminds us that saving species is ultimately saving ourselves. Each story is a mirror to human behavior and responsibility. The value of biodiversity. So the book opens with the provocative question, do we need every species? Is challenging us to reflect not just on ecological utility, but on the beauty, the meaning, and the richness each species brings to our planet. One of the key tools uh, we use today to assess the state of biodiversity is the IUCN Red List of Threatened Species. It is the most comprehensive global inventory of the conservation status of species in the world. The Red List currently includes assessments for over 160,000 species, and more than 44,000 of those are threatened with extinction, categorized either as critically endangered, or vulnerable. Since the year 1500, the IUCN estimates that more than 900 species have gone extinct. However, this is likely an undercount. Many species go extinct before they are even discovered, especially small invertebrates, amphibians, and tropical plants. According to the Red List currently, as you can see in the table, the groups that are on top of the list are the cycads with 71% of the species endangered, followed by amphibians. 41% of them are endangered. These are the most threatened vertebrates currently endangered in the world. And then 37% of sharks and rays. Of course, there is also a level of threat in each of the groups, but this is a summary that basically shows us what's the status of biodiversity nowadays. Thousands of insects and plants are also under severe pressure, but there is not enough capacity and research to understand what's the status. These groups are represented vividly in Extinctopedia, from the axolotl and Atelopus harlequin frogs to reef corals and pangolins, emphasizing both their uniqueness and fragility. The Red List doesn't just track losses, it serves as a barometer of conservation progress. It helps governments, NGOs, and researchers prioritize action, allocate resources, and monitor the effectiveness of interventions. The urgency is clear. We're in the midst of the sixth mass extinction driven by human activities mostly. But data from the Red List also shows that conservation works. Species have improved status due to habitat protection, breeding programs, and legal protections. So Extinctopedia invites us to consider what is lost when a species disappears, not only ecologically, but also culturally, scientifically, and spiritually. So we might wonder what are those drivers of extinction. Extinctopedia shows page after page how diverse and often overlapping human-driven threats have led to this disappearance of species. It makes abstract threats tangible and emotional. The main drivers of extinctions, as recognized by the IUCN and the Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Services, IPBES, include: first, habitat loss and degradation, the most widespread threat. Forests cleared for agriculture, wetlands drained, and coastlines urbanized. As an example, I can provide the many Atelopus frogs or harlequin frog species in the book lost to habitat destruction. Then we have overexploitation, hunting, fishing, and harvesting beyond sustainable levels. Martha, the passenger pigeon, is a clear example. Once the most abundant bird on Earth, wiped out by market hunting. Then we have pollution, agricultural runoff, pesticides, plastics, and industrial waste affect both land and aquatic species. Amphibians, for example, with their permeable skin, are especially vulnerable to this. Climate change alters habitats, shifts temperatures regimens, disrupts breeding cycles, and leads to phenomena like coral bleachings. Bleachings. Coral species mentioned in Extinctopedia, like the Acropora group, are at severe risk. Wildlife trade and traditional medicine, tigers and pangolins, prominently featured in the book, are severely impacted by illegal trade. Other threats include infectious disease. For example, the devastating fungal disease chytridomycosis that's wiping out entire amphibian populations and species from the face of the earth. And invasive species introduce predators, competitors, and pathogens wreak havoc on native species. So the interplay between these threats is accelerating the rate of extinction to unprecedented levels. For example, climate change can enable invasive species to thrive or weaken species' immune systems, making them more susceptible to disease. So many of these threats are reversible or at least manageable with effective conservation strategies, but action must be proactive and not reactive. Extinctopedia reminds us of the layered and often avoidable causes of extinction. It's not just about natural loss, it's about human choices. And then we have Martha's story. Martha, for those who don't know, was the last known passenger pigeon, and she died on September 1st, 1914, at the Cincinnati Zoo. Her death marked the extinction of an entire species that once numbered in the billions, the most abundant bird in North America and possibly in the world. In the 1800s, passenger pigeons flew in flocks so massive they could darken the skies for hours. Their social nesting behavior made them especially vulnerable to mass capture by hunters. They were hunted for meat, used as livestock feed, and their forest nesting grounds were destroyed. Despite early conservation warnings, no protective action was taken until it was too late. Martha lived the last years of her life in an enclosure at the Cincinnati Zoo. She became a public attraction, but also a symbol of loss. Her lonely death sparked one of the earliest national conservation about conversations, about the extinction, and helped fuel the modern conservation movement. After her death, Martha's body was packed in a block of ice and sent to the Smithsonian Institution, where she was taxidermed and placed on display. Today, she remains as one of the most powerful educational specimens in the museum's collection. Martha is featured prominently in Extinctopedia, reminding all of us how human indifference, not malice, is often behind extinction. Her story is a cautionary tale, but also a scientific legacy. Today, several organizations, including the Smithsonian and other organizations such as Revive and Restore, are exploring whether genomic data extracted from Martha's preserved tissues could one day be used in the de-extinction or genetic restoration project for the species. While fully bringing back the passenger pigeon is still far off and ethically complex, its DNA could be introduced into the genome of its closest living relatives, the band-tailed pigeon, to recreate traits or even a hybrid form. This work also helps refine biobanking, genome editing, and cloning techniques that are already benefiting critically endangered species, like the black-footed ferret and the Peruaski horse. Martha's lives on not only as a lesson, but as a possible bridge between the past and the future of conservation. And a story of hope, the black-footed ferret, almost but not extinct. So the black-footed ferret is a small carnivore mammal native to the Great Plains of the United States. It is North America's only native ferret species and a specialist predator, with about 90% of its diet made up of prairie dogs. By the 1970s, it was believed to be extinct in the wild, primarily due to two causes. The first one, widespread eradication of prairie dogs, as they were seen as pests by ranchers, and this led to the collapse of the primary food source and, and habitat. The second cause, disease outbreaks, especially sylvatic plague and the canine distemper, decimated both the ferrets and the prairie dogs. So in 1981, a small population of black-footed ferrets was rediscovered near Medici, Wyoming, by a ranch dog named Shep. This rediscovery shocked the conservation world and provided the last opportunity to save the species. So by the mid 1980s, only 18 individuals remained in the wild, and these were all brought into captivity to form the foundation of an emergency breeding program. The species was declared extinct in the wild shortly thereafter. Intensive conservation efforts followed, led by the U.S. Fish and Wildlife Services, with the key support from organizations like the Smithsonian Conservation Biology Institute, the Phoenix Zoo, Cheyenne Zoo, and other organizations, and a captive breeding, genetic management, disease control, and reproduction program started to protect the prairie habitats that form the pillars of the recovery plan. Reintroduction sites now span across several U.S. states, including Wyoming, South Dakota, Arizona, and Montana, as well as Canada and Mexico. As of 2024, the wild adult population is estimated to be 206 mature individuals, with over 500 animals in total in the captive populations. While still vulnerable, these numbers allow the species to be officially downlisted from extinct in the wild to endangered on the IUCN Red List. In a landmark conservation breakthrough, the first cloned black-footed ferret named Elizabeth Ann was born in 2020 using preserved cells from a 1988 individual named Willa. This work was done in partnership with Revive and Restore via GenPets, Equine, SCBI, and U.S. Fish and Wildlife Services. Cloning aims to restore lost genetic diversity and create a more resilient population. Willa's genes are now represented in the current wild living populations, so this effort helps counteract the effect of inbreeding and the bottleneck affecting species. The black-footed ferret story is one of the most successful examples of integrated conservation, where captive breeding, field ecology, genetics, disease science, and now cutting-edge biotechnologies all together playing a role. Featured in Extinctopedia, the near loss and recovery serve as a model for other endangered species and a reminder that extinction can sometimes be reversed with commitment, collaboration, and innovation. So that takes us to de-extinction. So what's de-extinction? It basically refers to the scientific pursuit of bringing back extinct species, or at least restoring their traits and ecological roles using modern biotechnology. Several high-profile initiatives are underway, and you probably are aware of some of those. The woolly mammoth. This project is led by a group of organizations like Colossal Bioscience, aim to create a cold-adapted Asian elephant with mammoth traits using CRISPR, gene editing, and Asian DNA. The thylacine, or the Tasmanian tiger, some efforts from the University of Melbourne aim to recreate this extinct marsupial using stem cells and marsupial surrogates from closely related species. And we already talked about Martha, the passenger pigeon, as well. So the main biotechnological tools being explored include cloning, using preserved cells from extinct or functionally extinct individuals, genome editing, such as CRISPR, Cas9, inserting key genes from extinct species into living relatives, backbreeding, and cryobanking and biobanking, which is preserving live viable tissue cells, gametes, and DNA to enable future applications. So while controversial, the extinction efforts also advance tools for conservation, including for endangered species, such as the genetic rescue, disease resistance, and assisted reproduction. Museums and herbariums provide vital DNA sources and tissue samples because these are living archives of what once was. So Extinctopedia raises the emotional and ethical stakes of extinction. The extinction asks by itself, if we could bring them back, should we first? And if we have the power to bring them back, do we also have the responsibility? What ecosystems would they return to? So, just for consideration. There are other stories of hope. Those species that were currently thought to be extinct, but they were rediscovered, as is the case of the starry night harlequin toad. So Extinctopedia reminds us that some species declared extinct have later been rediscovered, often in remote, hard to access habitats. And these are sometimes called the Lazarus species. And they symbolize resilience and the importance of never giving up on a species too soon. Uh, One of the cases, as I mentioned, is the starry night harlequin toad, rediscovered in 2019 in the Sierra Nevada de Santa Marta in Colombia, after being unseen for over 30 years. The rediscovery was only possible thanks to the local indigenous communities who had continued to observe the species but kept it protected in part due to cultural values and sacred beliefs. Their collaboration with scientists and conservationists demonstrated the power of community-based conservation efforts. The toad's survival is attributed to its isolation because it's located in high altitude habitats, and this might shielded it from the chytrid fungus. This rediscovery gave new momentum to similar efforts across Latin America, including the ongoing search for the Panamanian golden frog, which is a national symbol of Panama that is believed to be extinct in the wild due to the chytrid fungus. Since 2025, a coordinated effort to search for surviving wild populations has been launched, supported by a coalition of partners, including ReWild, the Amphibian Survival Alliance, the Smithsonian Tropical Research Institute, and the Bezos Earth Fund. These efforts use camera traps, environmental DNA, sampling from streams, and close collaborations with local communities to detect remaining individuals. Even if no wild populations are found, this is building momentum for future reintroductions from assurance colonies or captive breeding programs, mainly the one from Panama Amphibian Rescue and Conservation Project. So basically what we're trying to show you in this slide is the fine line between extinction and survival, and the enormous impact that collaborative, hope-driven science can have on conservation outcomes. These amphibians, though small and fragile, serve as ambassadors for entire ecosystems, reminding us that while extinction is final, rediscovery is always possible. And then we have a successful story, the conservation efforts on the panda, the giant panda. And this is one of the most iconic conservation success stories of our time, probably. It was once a global symbol of extinction, and now it's a symbol of hope. In the 1980s, panda populations in the wild were declining rapidly due to habitat fragmentation, low birth rates, and limited food sources. It's important to mention that they feed almost exclusively on bamboo from bamboo forests. So fewer than 1,000 pandas were estimated to remain in the wild. So responding to this crisis, the Chinese government, together with global partners, initiated one of the most coordinated conservation efforts in history. So this included the creation of over 60 panda reserves across China, strict anti-poaching laws, and bamboo forest protection and restoration, advanced captive breeding programs with an emphasis on genetics, behavior, and health. The Smithsonian's National Zoo and Conservation Biology Institute played a key role in this effort with their captive breeding program and breakthroughs, including artificial insemination and cub development monitoring, pioneering research on panda nutrition, reproductive cycles, and maternal behavior, and supporting field conservation and capacity building with partners in China. So in 2016, after decades of efforts, the giant panda was officially downlisted from endangered to vulnerable on the IUCN Red List, a rare and celebrated reversal of an extinction trend. This story underscores what's possible when science, Diplomacy, long-term funding, and public support align. As Extinctopedia shows, extinction is real, but also is recovery. The panda teaches us that we, with commitment, even with the most fragile species, we can bring them back from extinction. So now I'm going to turn it to Brian, who's going to talk about tigers.
Thanks so much, Gina. We're running a little behind now, so I'm going to try and get through this quickly. I started my career in conservation as assistant director of Save the Tiger Fund, which was trying to give out money to people working in 13 tiger range countries to try and recover tiger populations. And that was a really important and formative piece of my conservation education, because it was there I got to interact with experts, some of the tiger wallahs of the day, the John Seidensticker, AJT Johnson, Ulas Karanth, these tiger experts working day in, day out to save tigers and to rebuild what they call the tiger conservation landscape, right? So that these tigers can move between little tiny reserves and spread their genes and manage their full potential as a species. And it was there that I really learned that it's not just science that saves species, it's direct conservation action. And as conservationists, we have a limited set of tools that we can all use to save species. Those include species conservation actions. They include setting aside habitat for conservation and education. Next slide, please. I also, before I was at Save the Tiger Fund, was a marine biologist working on coral reefs in the British Virgin Islands. And at that time, there was about 10% of the reefs were covered in live coral. And I read in the books about how the days in the Caribbean before the die-off of the long-spined sea urchins, that there was 40 to 60% live cover coral in the Caribbean. And I imagined those days and thought, wouldn't it be spectacular to see reefs like this? And I went just before COVID back to the British Virgin Islands, and that coral cover had dropped from about 10% to less than 1%. And so corals are dying out. Climate change is a big problem with that, leading to a lot of bleaching. My colleague here at the Smithsonian Conservation Biology Institute, Mary Hagedorn, is trying to cryopreserve cells of these corals and embryos so that we can store these corals genetically over the longer term. And she's also been working on the process called isochoric vitrification, where you can take a little chunk of coral and freeze it and thaw it for future use, because we really are on the brink with a lot of these species. And this is one of our last options, is to use this technology to preserve some of the genetic diversity of this species. Next, Gina. So there's also a lot of other species that I've touched on in my career. I have some colleagues here at the Smithsonian who are working on turtle conservation. And we have a very rare turtle in Virginia, the wood turtle. And here you see some of our colleagues out there. They count all of the turtles that overwinter in the streams every year and try and monitor them to see how they're doing and to see if we can prevent this going onto the endangered species list. But there's also, we've got a lot of work to do, because there's about 500 species of turtles and about half of them are endangered. And we're also working globally with the Turtle Survival Alliance, and that the mission of that organization is to prevent all turtle extinctions. My sister-in-law works in Zimbabwe, and she works with the Tiki Highwood Trust, and she rescues pangolins that have been confiscated from poachers. And if the pangolin mother has a baby, she will have to hand rear that. So it's a very important amount of work that she's doing. And axolotls, that is a very interesting species, because it's almost extinct in the wild, because Mexico City was built on top of Lake Xochimilco, which is the home of the axolotl. Yet it's one of the most popular and widespread pets in the world. So, there is an opportunity to now release captive bred animals back into the Lake Xochimilco. Next, Gina. So we also work in museums, and apart from having like preserved specimens of Martha the pigeon, we have been working on a lot of technologies to try and maximize the value of some of these collections. So Gina herself has been working on special cryopreservation protocols to collect and freeze living sperm of these endangered amphibians so that we can thaw it and use those genes in the future. There's also technologies where we're freezing cell lines. We try and get cell lines into a culture medium that can be cryopreserved and thawed at a later stage. And so that way, we're not going back to, specimens where the DNA has been very destroyed through freezing or formalin preservation, and we have really good, high-quality living DNA to work with as part of these sort of de-extinction technologies. Next, Gina. So my conclusion is we study this sort of gradient of extinction, right? From a healthy species in the wild to completely extinct. And there's also, various different steps along the path to get there. And part of that is understanding how threatened these animals are. But there's also a large amount that we can do as conservationists to take steps to ensure that these incredible species will hang around for the future. And I am a member of the Amphibian Survival Alliance that Gina leads, that helps support global capacity building in conservation. And if you want to help amphibians, I would say that that's a really good first step, is to join the Amphibian Survival Alliance. Next, Gina. And so I want to thank the author, Serenela Quarello, and the beautiful illustrations, Alessio Alcini. It's a beautiful book, and this kind of communication, this kind of education is really important for us to get the word out about extinctions and to get people to care. Thank you. Next, Gina. All right, and now we're going to switch gears a little bit and talk more about our work. So, Gina, can you load up our...
I need to change the presentation. Hold on.
So to occupy the time, Gina and I both work on an amphibian conservation project called the Panama Amphibian Rescue and Conservation Project. And it is an effort to create captive assurance colonies of highly endangered frogs in Panama that are threatened with extinction. So the current situation in the neotropics is amphibians are highly threatened or endangered, and the hotspots of biodiversity you see here on this map are the tropical Andes and all through Middle America. About 41% of amphibians are threatened or endangered, and we've also lost a lot. Just in Panama alone, we've lost 9 species of amphibians, including Adelopus chiriquiensis, which is shown here. Next, Gina. So the Panama Amphibian Rescue and Conservation Project, we literally built an amphibian ark where we are breeding endangered species in captivity. And we're also trying to figure out a solution for the amphibian chytrid fungus for reintroducing them into the wild. Next, Gina. So our facility, we took these shipping containers. I saw all of these shipping containers moving up and down the Panama Canal. And I thought, well, if we could get some of those shipping containers and convert them into little zoos where we can keep and breed these frogs, we can keep the disease out and keep the frogs safe inside. So we have 7 of these containers that are filled with with 9 different species of frogs. And we also have an insect breeding facility and offices for our staff. Next, Gina. So these are some of the beautiful species we are working to breed in captivity. We have about 2,000 adult frogs, and they are some of the last of their kind. Just very, very beautiful species. And these ones are amongst the most at risk of extinction in Panama because of the amphibian chytrid fungus that's wiping them out. And in particular, the group that Gina mentioned earlier in the presentation is this harlequin frogs, these very beautiful, colorful Atelopus species, and they are some of the most at risk from the amphibian chytrid fungus. Next, Gina. We also do active exhibitions to show people and to communicate the value of frogs. And what you see here in this lower image is our center at the Punta Culebra Nature Center. And it's a very, very beautiful exhibition. But what it's really showing you is that if you can show people how much you value frogs by providing them an extraordinary level of care and attention to detail, that the real message that frogs matter and frogs are important is really well communicated. And our center at Punta Culebra is visited by about 20,000 school kids every year. Next, Gina. So our research priorities. I've spent a large amount of my career really trying to figure out solutions for the amphibian chytrid fungus. This has included trying to find protective skin bacteria that produce antifungal chemicals that might confer protection on threatened frogs. We've also tried developing vaccines to expose frogs to the chytrid fungus and then clear them and then re-expose them to see if that helps. And also trying to breed resistant individuals. We haven't really found a whole load of promising results in those efforts. Vaccinating some of these harlequin frogs actually makes them more susceptible to the disease. And the probiotics, we even tried genetically engineering a bacterium that's very common on golden frogs and putting it back on the frog, and it actually did not help save the frog from the chytrid fungus. Next, Gina. So what we're doing right now is some reintroductions, and we're trying several different ways to see if we can put our thumb on the scale in favor of the frog. One is this fairly common dart frog species, Cholesthetus pradi. It disappeared from a national park where we're working completely because of the chytrid fungus, but it did recover on some other mountains. And we went out with Gina to go and collect a founding population of 12 pairs of animals. And we're going to try translocate that to the exact site where we knew they occurred previously, and see if we can translocate a resistant individual. The middle frog is the lemur leaf frog, and here you see Julie Duggar introducing some of these tadpoles into big, like, cattle tanks we have out in the rainforest. And she's been putting some antifungal medicine in some of those cattle tanks to see if we can help improve the survivorship of those tadpoles and help build those populations of lemur leaf frogs. And then finally, that last frog, the crown tree frog, it has a radio transmitter tied to his back. And we will be putting those in artificial tree holes where they breed in nature, and see if we can track those captive bred individuals and see how they do. Next. While we were out working on collecting, actually looking for the Panamanian golden frog, we did encounter this species, the Vicente's poison dart frog, and we encountered it from that red dot on the map where it was thought to be extinct. So we have actually found these using audio moths, which are little devices we put out in the rainforest and record 1 minute every 10 minutes, and then we can use AI to do pattern matching analysis and see if we can detect the frog calls of species concerned. So that is some hope. We've been able to use this 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 audio moths and monitoring of frog calls is a really important tool for conservation. Next, please, Gina. And this is where I'm going to hand it over to Gina, because she's the master at assisted reproductive technologies.
Thank you, Brian. So under our research priorities, the development of assisted reproductive technologies and biobanking is it's included. And these are basically aimed to develop safe protocols to keep the captive breeding populations going and to safeguard their genetic variability. On this picture here, amazing picture taken by Brian, actually, you can see one of the harlequin toads, Atelopus varius. This is a post-metamorph created or produced by artificial fertilization. So these are very, very successful ways to enrich and to complement the other captive breeding management tools that are used on captive breeding programs. And I also want to kind of present to you what comes from meaningful collaborations, and in this case, the collaboration from between the Smithsonian and the Amphibian Survival Alliance. But first, I'm going to talk to you a little bit about what ASA is. And basically, we're a partnership of currently 86 organizations around the world, very committed and working on amphibian conservation actions on the ground. And our vision is to build a future where amphibians thrive in nature. And we do this working in close relationship with other two sister organizations, the IUCN Amphibian Specialist Group and Amphibian ARC. And the three of us share the same vision, which is bringing back amphibians and mitigating extinction threats. And then also, since we're on a library talk today, it's important to know that there are many very meaningful documents out there that guide our conservation actions. One of them is the Amphibian Conservation Action Plan, recently published in 2024, and the State of the World's Amphibians, which is the result of the second Global Amphibian Assessment published in 2023. And this assessment was the one that gave us that 41% number of threatened amphibians globally. And the Amphibian Conservation Action Plan, it's a global plan. It has more than 100 authors, this amazing document, and it's a guide for conservation actions for amphibians. And this is divided, let's say, in the main threats, the decision-making tools and management strategies, and gives basic but pivotal recommendations for amphibian conservation. So ASA guides global conservation actions using these two, the latest documents and data as a roadmap. And we also have several priorities, but I would say the main one is strengthening the partnership for greater impact on the ground. So what we do basically is we support this, the partnership and the initiatives through several strategies, including fundraising, providing funding and facilitating resources, supporting action on the ground, integrating policies, providing capacity building, and fostering collaborations. So this is a map of where our partners are located. We're growing at the moment. And these are some of the organizations that are part of the partnership. And as you can see, there are like big organizations working in several groups, taxonomical groups, and there are others that are amphibian focused. But I think the main and common theme here is organizations are committed on amphibian conservation among their priorities. And one of the examples of what ASA does and can do is this recent regional strategy that we developed in collaboration with Smithsonian, and that's supported by the Bezos Earth Fund. And this is an initiative that includes 4 countries: Ecuador, Colombia, Venezuela, and Panama. And it's tackling several amphibian conservation priorities in each of the countries to make it a regional strategy, including strengthening the national conservation action plans for amphibians, also developing biobanking hubs and capacities for each of the countries, developing work in situ and ex situ, strengthening the ex situ capacities as well, And we hope to impact at least 65 species by the end of the project. So this is an example of what ASA does. We kind of identify opportunities, bring together our partners, and coordinate local, regional, and global efforts. These are some of our supporters for both PARC and mainly PARC and ASA. And we want to thank you for your attention. And I want you to pay attention to the image on the right of your screen, because this is what will happen if frogs will go extinct and we don't even realize it. So thank you.
Thank you both so much. Yeah, the picture on the right of the screen gave me chills. We don't think about it. I never thought about that. So thank you for sharing that. Okay, so we have a lot of great questions. I'm going to dive in first with Ivy's question. Ivy from East Brunswick Public Library says, what role do invasive species play in biodiversity loss? And are there examples from your own research or from the book that could illustrate this impact?
Yes, of course. Invasive species is one of the main threats, for example, to amphibians. And one example I can give you is trout. It's a species that's usually brought to new environments where it was not originally from, and they feed on eggs and tadpoles, and then they have decimated enormously and impacted many populations and species of amphibians. So yeah, introduce an invasive species because it's kind of the same. Some introduced species are not invasive. Some invasive are also introduced. So they play a main role on it. I don't know, Brian, if you have any.
The amphibian chytrid fungus is also considered an invasive species because it is thought to have originated in Southeast Asia, and the global pandemic lineage has spread and caused particular devastation in the New World, in the Americas and Australia and New Zealand. And that's just where there's just no evolved resistance to this pathogen. I also worked, in Zimbabwe, and we have largemouth bass from the United States are horrible invasive species there. And they're associated with an average of a 99% reduction in your native minnow population. And they also predate on tadpoles.
Wow. 99% reduction. Wow. So Gina, you started to touch on the reintroduction could also be considered invasive species. And Andrew is asking a question about that. Reintroducing an animal that is visually similar to an extinct species has other implications. Is the newly designed animal susceptible to disease in the same way? And then does it just become an invasive species itself?
No, there's a difference between reintroduction and introduced species. So reintroduction is when we introduce back into a habitat, the species that used to be there. So it has an ecological role already in that habitat. An introduced species is a species that's foreign to that specific habitat, and we're introducing that species that didn't belong there with its own ecological role impacting that already established ecological role that existed without that new species. So in terms of reintroduction, could you please read the question again? Because I wanted to make it clear first.
Reintroducing an animal that is visually similar to an extinct species has other implications. Is the newly designed animal susceptible to disease, and will it also just become an invasive species itself?
So he's talking about de-extinction and bringing back something similar that's not quite the same species. Well, we don't know that. And actually, that's one of the questions that we need to consider before making those decisions. I think, and it's my opinion, that if one of these projects is successful, creating individuals that are either similarly genetically speaking, similar to those species, or bringing back the actual species, they will first have to go to captivity, to captive environments to see what happens, what are their needs, how they behave in terms of needs and physiologically speaking. And then after that, if even possible, thinking of reintroducing them. But I don't know, Brian, if you have another view on it or.
No. You said in your opinion, so the scientific community isn't necessarily in agreement on that?
Yeah, there are different views on de-extinction and what to do with whatever is de-extincted.
Mm-hmm.
Yeah, but it has so many benefits in different aspects. For example, the actual technologies that are being developed with that objective are applicable in many other aspects for conservation, not necessarily for de-extinction only. So in terms of technological development, it's great. But then again, we need to be very careful in terms of what we bring back if we do it. What species, if they still have a role in the ecosystems or not. I think there's a lot of debate and discussion going on, and that will continue happening in the near future.
Okay. So we've had several people ask about the fungus that you were talking about with killing the frogs. Lynn says, is this fungus worldwide and could it be changed genetically to keep from killing the frogs? So, and other people have said, can we do something about the fungus instead of the work on the frogs? So what does that research look like?
So we've been this chytrid fungus. There's two species that we know of: the salamander-eating chytrid fungus, and then the regular amphibian chytrid fungus. The regular amphibian chytrid fungus has several strains, and the one that's affecting us in Panama is called the global pandemic lineage, and it's quite virulent. There are other less virulent strains of the chytrid fungus out there, but that does not help the amphibians to form immunity to the disease. We found that even if you expose an amphibian to a less virulent strain, when you expose them to the more virulent strain, it's just double trouble. So that's one of the issues. Some people have found that some species do acquire some immune response to the amphibian chytrid fungus, and if they see it a second time, they would be more able to resist it. So there's a researcher in Australia, Anthony Waddell. He found that these frogs can clear the amphibian chytrid fungus through heat, and he builds like little greenhouses with bricks inside that the frogs like to hide in, and it also heats up the habitat enough that the amphibians will clear the amphibian chytrid fungus. And then they also seem to have an acquired immune response, so that if they see it again, they will be able to better fight it. But we did try that experiment with some of the harlequin frogs to see if we could clear the amphibian chytrid fungus and then re-expose them, and we found they were actually more susceptible the second time we exposed them.
Oh no.
I think it would be worth also to give a little bit of background on the fungus. So for people that are not very familiar, this is it originated, as Brian mentioned, on the Korean Peninsula, and there are many theories on how it spread all over the world. And some of those are first due to the Korean War. So all the logistics and movement that happened in the area helped transmit all the spores everywhere. And another theory that is probably complementary is the use of frogs and toads as pregnancy tests early on during the 1950s. So hospitals and laboratories will use frogs and toads to do these pregnancy tests. And these were the precursors of the current rapid home test that we have. And they will basically take urine from the woman that was suspecting pregnancy and inject it in the abdominal cavity of the frog, wait for 2-3 days, and after 2-3 days, they collected urine, and if there was sperm in the urine, it meant that the woman was pregnant, because it stimulated the male with the reproductive hormone that's in the urine of the female, of the
Wow.
woman. And then after a while, technology kind of progressed, and the new tests were developed, and many frogs were also released, even though they were not local, because they were many of them were introduced from Africa, and these are resistant species to the fungus. So yeah, there's many different theories in terms of how it's spreaded. And the other thing is that once the fungus attaches to the skin, because it will affect the skin of the amphibian, it will basically thicken the skin, and the animal will die from cardiorespiratory arrest.
Wow.
The way it reproduces is the fungus that produces spores that are motile, so they swim in the water until they find another frog and attach to them. So in terms of, there are some initiatives trying to genetically modify the fungus. For example, there were a couple of ideas trying to genetically modify the molecules that control the tail of the spore so they couldn't swim. The thing is the scale. Think about every river in the world, every water body in the world with this. So it would be very complicated and take a lot of time. And additional to that, the strains Brian mentioned. There are different genetic strains of this fungus. So
yeah. Okay. Thank you for sharing that context. So Yasmin from Calgary Public Library has a question for each of you. I'll start with Gina's question. Assisted reproductive technologies are a fascinating and hopeful avenue. What are the biggest scientific or ethical challenges you've faced in applying these methods to critically endangered species?
Well, when we talked about artificial assisted reproductive technologies, we're mainly focusing on hormonal treatments to stimulate the production of eggs and sperm. We also talk about artificial insemination for those species that have internal fertilization, and artificial fertilization for those who have external fertilization, such as frogs. We also work on ICSI, which is intracytoplasmic sperm injection. So those usually don't have many ethical concerns. Actually, we think of those as a responsibility when we have a species that's perishing in the wild, and we have the last individuals in captivity. So we help them reproduce so they don't go extinct.
Mm-hmm.
But when you talk about advanced arts, in this case, for example, cloning, and these other techniques, it depends. I would say first on the institution, because every institution has, or usually they have a committee that oversees the ethical and, let's say, correct treatment of animals and procedures, and then also what's the objective of that.
Mm-hmm.
So when we're talking about arts and biobanking to support ex situ and in situ populations that are going extinct, this is usually what we refer to, and I wouldn't say there's ethical concerns, but then the other ones, and depending on how they're looking to be applied, might be subject to probably scrutiny or a little bit more of questioning within the institutions and the research collaborators.
Mm-hmm. Okay, thank you. And Yasmin's question for Brian says, given the dual threats of disease and climate change on amphibians, which conservation strategies do you see as most scalable and effective for long-term success?
I think the most scalable conservation intervention for amphibians is to give them places where they can breed and reproduce. I have in my backyard in Virginia, I've dug 10 little vernal pools that there's no fish in, and they are filled. They're just little amphibian factories pumping out baby amphibians onto the landscape in vast numbers. And when I work in the Panama Amphibian Rescue and Conservation Center, we have to grow all of the food for these animals. We have to manage all of the care. In some cases, Gina has to help them reproduce with hormones. It's very expensive. It's very difficult to recreate nature inside a building. And the best thing you can do is to help create opportunities for amphibians in your own backyard. And I just think that that's the most scalable solution. When you give them fishless habitats, they will come and they will reproduce. And about 4% of amphibians in the United States are disappearing every year. And largely because of this habitat issue, right? There's just they need these vernal pools, these seasonal pools that will dry up in the summer to breed and to thrive. And the reason they can breed and thrive there is because there's no fish. Yet a lot of people will see these vernal pools and they say, "Oh, mosquitoes, we need to drain that." And they'll dig a ditch and then the vernal pool will be gone. So, even if you can just do a little backyard pond with no fishes in it, the amphibians will find it. And once the amphibians find it, they will take care of the mosquitoes.
Oh, see, I like that.
Can I add something else?
Sure.
In terms of, because Brian is referring a lot to kind of North American amphibians, but the scenario is different in the tropics, for example. And I would say one of the structures that we've seen that can work, it's habitat restoration, but that needs to be a country level priority that need to be pushed in collaboration between research organizations, government organizations, civil society, on the basis of education. So I would say starting with educating the community for them to understand what's at risk. And that facilitates then the development and implementation of different strategies. So for climate change and disease, give them space, as Brian mentioned, to breed and to be. So restoring their habitats, the wetlands, for example.
Okay. Well, great. That is unfortunately all the time that we have today. Thank you both so much for your time. What an incredibly informative hour. And I can't thank you enough for being here.
Thank you for having us. And thank you, everybody, for being here.
I want to echo that, extend a huge thank you to all of our audience members for joining us. Without your support, none of this would be possible. And we thank you for choosing to spend your time with us. Below the live stream video in the resources area, you can find a link to the book and to ASA, as was mentioned with Brian and Gina's research. We look forward to everyone joining us next week on June 10th for an afternoon of Thrill, rather, as we chat with New York Times bestselling author Lisa Jewell. You can be sure to register for this and lots of other great events on your library's event page. I am Brandon Adler, and thank you for joining us.